All right. Well, welcome everyone. Let's get started today. So it's my pleasure as the theme leader of the regenerative biology and tissue engineering theme to be hosting Professor Stacey Finley today in our theme virtually. She's joining us from University of Southern California, where she's an associate professor and the Gordon Marshall Early Career Chair in Biomedical Engineering at USC. I've known Stacey for quite a long time. She's been at USC for almost a decade after doing an undergraduate degree in Florida A&M in chemical engineering, graduate work at Northwestern in chemical engineering, a postdoc at Johns Hopkins, and then being almost a decade at USC and really doing exceptional work at thinking about ways to apply computational methods to a lot of really complex systems. Along the way, she's been recognized by a lot of different awards and different programs, including an NSF Career Award, and last year being elected as a fellow to AIMBE, which is meant to represent the top 2% of medical and bioengineers in the country. And so it's a real honor to have Dr. Finley join us today to talk about computational modeling of the tumor ecosystem. So I'm going to turn it over to Dr. Finley. If you have questions, feel free to put them in the chat or save them for the end, and we'll have a question and answer period at that point. So, Dr. Finley.
Great. Thanks. Thanks so much, Brendan, for the invitation and the kind introduction. I'm excited to be able to tell you about some of the work that my lab is doing. I'm a little bit disappointed that I couldn't be there to share in person, but happy to, answer questions afterwards and also talk with people if you're interested in some of the things that come up. So what we're really looking at in my lab is more broadly situated in the field called mathematical oncology. Studying cancer with mathematical and computational tools, I think, is quite appropriate because of the complexity of cancer, because of the different processes that are involved, such as angiogenesis, the formation of new blood vessels, of course, the different cell types that are there, including tumor cells, but also immune cells and other stromal cells, and how all of these cell types and processes evolve and the dynamics of them changing over time. It leads to some very complex morphologies of the tumor and also impact in terms of prognosis and the response to treatment. And so we have been using mathematical models, specifically mechanistically detailed models, to better understand cancer. And so the main goal of my research lab is to be able to predict the dynamics of networks in cancer that could help us to understand how vulnerable those networks are so that they can be exploited to ultimately inhibit tumor growth. And we do this using systems biology tools. So that means studying the whole instead of individual parts. So not just one reaction or not just one molecular species, but how those reactions and species are involved in a larger network and give rise to system level behavior. And we can do that using mathematical modeling, which is the expertise of my lab. And we lean quite closely on experimental collaborators to provide data for building the models and also validating the model predictions. So the three main areas of research in the lab are to look at cell-cell interactions, which I'll talk about in the second half of the talk, to look at tumor angiogenesis, which stems from some of the work that I did as a postdoc and starting early on as a faculty at USC, and then also cancer metabolism. And so I'll start here with cancer metabolism because we have some new work that's come out of the lab that we're quite excited about, and then move into the area of cell-cell interactions. But all of this really has to do with what's happening in the tumor microenvironment, how are cells interacting with one another, and can we understand those interactions enough to be able to exploit them for inhibiting tumor growth. So in terms of cancer metabolism, we're really focused on colorectal cancer, and there are a number of reasons to look at this particular type of cancer. It's the second leading cause of cancer-related deaths in men. The 5-year survival rate is quite low, particularly for metastatic disease. There are just a few limited treatment options for tumors that have this mutation in KRAS, which is about 90% of colorectal cancers. And then that gives rise to resistance to standard therapies, and that certainly contributes to the morbidity and the mortality rates. So one aspect that is quite tightly, I think, integrated into the response to treatment and really the resistance to treatment are what's happening in the microenvironment. And we've particularly gotten excited about the role of cancer-associated fibroblasts, or CAFs, in the local microenvironment, which can play a whole host of different roles. So they can lead to education of genotype-driven education of the both cancer cells and CAFs. So this bidirectional really relationship between them, leading to epigenetic reprogramming, influencing the spatial distribution and organization of the tumors, promoting stemness in cancer cells, and then also influencing the metabolic reprogramming. And that's what we're really focusing in here on this project, is what are the interactions, the metabolic interactions between cancer cells and cancer-associated fibroblasts, and they certainly influence the response to treatment. So we have gotten really excited about being able to study the metabolic effects of cancer-associated fibroblasts and how they influence the metabolism of colorectal cancer cells. And to do this, we worked with Shannon Mumenthaler's lab, and a PhD scientist in her lab, Dr. Emma Fong, was able to set up this series of experiments that I'll describe to you, and how we can use that as a platform to study metabolic interactions between CAFs and colorectal cancer cells. So what Emma did is to culture colorectal cancer cells in the media that they're normally grown in for 24 hours, and then replenish the media after another 24 hours. So we go from T equals 0, which is when the media would be replenished, out to T equals 24 hours. And at the same time, she can first start the cells in their CRC media, and then switch the cells to media that's been conditioned by CAFs. So by this, I mean separately, she had cultured CAFs for about 72 hours, and then taken the media that they were growing in and used that now to culture the colorectal cancer cells in. And so that is then allowed to move forward for 24 hours. And so we have these two time points and these two conditions where colorectal cancer cells are staying in their own CRC media, or where they're switched to CAF conditioned media. So those are the two conditions that we have. And then we move forward with a collaboration with Nicholas Graham, who's also at USC, in order to measure, using mass spectrometry-based metabolomics, to measure the intracellular metabolites inside of the cells for these two conditions, and specifically quantify the fold change in the metabolite levels for 24 hours relative to the 0-hour time point. And then with those metabolomics data, then we can move forward with the kind of modeling that my lab has expertise in, namely constraint-based modeling, where we can look at the flux through these metabolic pathways. And this is with a former postdoc in the lab, Dr. Junmin Wang. And so the question that we wanted to answer here is, what are the effects of the CAF conditioned media on colorectal cancer cell metabolism? How do CAFs influence the cancer cell metabolism? What's the reprogramming that happens due to the cells being grown in this conditioned media? So the very first thing that we looked at is, well, what's happening to the cell morphology and also the cell growth rate? We were finding that when the cells were retained in their colorectal cancer media versus those that were switched to CAF conditioned media, it does change the morphology. And so what I'm showing here are the results from our studies with the DLD1 cell line that either has wild type KRAS or has the KRAS mutation. And so across the two rows are those two cell lines, and then across the columns are the different the different growth media. And so we can see that cells that are retained in colorectal cancer media remain rounder, and they have this morphology that's shown here, whereas where they're switched to the CAF conditioned media, they have more Filopodia-like structures, and they're elongated. And so we can certainly see that it affects the cell morphology. But interestingly, we didn't find any differences in the cell growth rate. And so I've color-coded these boxes to show the DLD1 cells that are having wild-type KRAS in the CRC only media is the light blue bar here. CAF conditioned media is a dark blue bar, and then the same light and dark for the cells that have the KRAS mutation. And you can see that there's very little difference in the growth rate. So we don't see at the bulk level changes in how fast the cells are growing just because they were grown now in the CAF conditioned media. So that led us to say, well, we don't see anything at this bulk level. Maybe we can go in and look at what's happening inside of the cells as a way to give us insight into how the CAF conditioned media might be affecting the colorectal cancer cells. And so that's where we started to look at, well, what is the effect of the CAF conditioned media on particular metabolism of the colorectal cancer cells? And what we could do is to make two different comparisons, right? We can compare the effect of the media. So those were cells that were grown in CRC only media versus those that were switched to CAF conditioned media. We can also compare the effect of the mutation, wild type versus KRAS mutant cells. So first, looking at the comparison of the conditioned media, the effect of the media, we can measure, again, with the metabolomics approach, what are the fold changes of certain metabolites that are inside of the cells. These are cells that do not have the KRAS mutant, and we can take the fold change of the relative metabolite levels so that we can compare the effect of CRC only media to the effect of the cells that were switched to CAF conditioned media. And again, this is just for wild type cells. We can see that based on statistical analysis, there are several metabolites that have a significant different abundance level when we're comparing CRC only media to CAF conditioned media, and those are the ones that are highlighted in red, and then we're just labeling those that are present in the central carbon metabolic pathways. So here there are about 25 metabolites that have differential abundance level just due to the CAF conditioned media. This is true for wild type. This is also true for cells that have the KRAS mutation. About 2 dozen metabolites have differential abundance when the cells have this mutation when they're switched to the CAF conditioned media. And this is in stark comparison if we're only looking at the effect of the mutation, so the genotype. So here we can look at those that were remaining in CRC only media, those were switched to CAF conditioned media, and there's just 5 or 2 metabolites that have a differential abundance. And so what this is telling us is that certainly the effect of the conditioned media is stronger than the effect of the mutation. And so we wanted to further probe what's happening due to the CAF conditioned media. So then we looked at, okay, what are those metabolites that are altered or having a differential abundance? And we can do that in a couple of ways. So we can, again, you're going to see this color scheme for the next several slides, where the blue corresponds to wild type cells, the green corresponds to cells that have the KRAS mutation, light colored bars will correspond to cells that were remaining in CRC only media, and the dark bars are cells that were switched to CAF conditioned media. So we can look at what is the relative fold change of the metabolite levels. We're always going to normalize them to what's happening in wild type cells with CRC only media. So you'll see this gray dashed line going across that will indicate relative to this particular condition. And so we can look at what's happening for different species in glycolysis pathway, in the PPP, pentose phosphate pathway, and then also the TCA cycle. And so what you should take away from these two figures, whether we're talking about wild type cells or KRAS mutant cells, is that there's really no coordinated change in the levels of the metabolites for a particular pathway. So they're increasing, they're decreasing, both within glycolysis, within pentose phosphate pathway, within the TCA cycle. So just looking at individual metabolites doesn't give us a lot of insight into what's happening in terms of the reprogramming of the colorectal cancer cells due to the CAF conditioned media. But we know, right, that within these reactions, these metabolites are being synthesized, they're being converted to other metabolites, are being used up. And so it's not sufficient to just look at individual metabolites. We really need to take a more systems level perspective and see, well, how are the metabolites actually interconnected through metabolic pathways? And so that is now what I'd like to talk about, is what are the fluxes through these metabolic reactions that simultaneously take into account substrates and products. And so what we're doing now is using a constraint-based modeling approach, which allows us to predict the fluxes through these metabolic reactions. We're focusing in on central carbon metabolic pathways, so glycolysis, pentose phosphate pathway, and the TCA cycle. And this is a computational analysis that allows us to predict the reaction fluxes. And so we have to have some inputs to this analysis. So we take the initial metabolite levels from the literature, we take our measurements from mass spectrometry-based metabolomics that tell us the fold changes of those metabolites over 24 hours. These provide some constraints on the mass balances within those reaction networks. We also have to specify some bounds about how much flux could go through the reactions. So we take directly from our mass spectrometry measurements the rate of nutrient uptake, so uptake of glucose, uptake of glutamine, and secretion of lactate. So those are fixed. And then we have bounds on the fluxes, whether they're reversible or irreversible. And then lastly, again, from one of those very initial slides that I showed, we have the measured growth rate. So for us, that means that we can further constrain the model to have the biomass production, which is within this network of metabolic reactions. That biomass production rate is exactly fixed to the growth rate that we measured. And with those bounds, we can predict what is the flux going through every reaction in our metabolic network. And we do this using unsteady state parsimonious flux balance analysis. And I'm happy to talk more about the details of this computational approach. But again, it allows us to predict how much flux is going through each of the metabolic reactions. There's a whole, iterative approach to estimating and predicting the fluxes, but what we get out are how much, what is really the reaction flux or the rate of each of these metabolic reactions, given these constraints on the mass balances. So we can not only look at how the individual metabolites are changing over time, but also how that influences flux through the reaction network. So we can start by looking at glycolysis, and I show the schematic here, but then we can look at what are the fluxes through each of the reactions in the glycolysis pathway. Again, just orienting you to what I'll be showing, the relative flux for cells that have a wild type KRAS, for cells that have mutant KRAS, and those that are remaining in CRC only media, so the light colored bars, versus those that are switched to CAF conditioned media. So for the first reaction in glycolysis, the hexokinase reaction, we see that the presence of CAF conditioned media in these wild type colorectal cancer cells increases the flux through that reaction. This increased flux through this glycolytic reaction is repeated for the other reactions in this pathway. So we see a coordinated increase in the flux through glycolysis just due to the presence of the cells being grown in the CAF conditioned media. Now, just to note that I'm highlighting in red the GLUT reaction, which is the uptake of glucose, and the MCT reaction, which is the secretion of lactate, because those aren't predicted by the model. They are fixed based on our experimental measurements. So we see this coordinated increase in the glycolytic fluxes, both for cells that have wild type KRAS and the cells that have mutant KRAS. So several fold increase in the fluxes for some of these reactions because the cells are now grown in the CAF conditioned media. So this is what's happening in glycolysis. We can also look at what's happening in the pentose phosphate pathway, both the oxidative arm of the pathway and the non-oxidative arm. So for oxidative pentose phosphate pathway, we also see a coordinated increase in the fluxes through those reactions. For non-oxidative arm, either there's no change or maybe a slight decrease in the flux when we're considering cells that have the KRAS mutation. So this helps us to understand. We know that the pentose phosphate pathway is important for producing reducing equivalents that are needed for producing other cellular resources for proliferation or biomass maintenance. And so it helps us to understand how these metabolites and nutrients are being used, particularly when the cells are switched to CAF conditioned media. We can also look at TCA cycle and glutaminolysis, so the utilization and metabolism of glutamine. For TCA cycle reactions, both for wild type and for KRAS mutant cells, there's a decrease in the flux through all of those reactions, well, nearly all, with the exception of the MDH reaction. And that's true for wild type and for KRAS mutants. So TCA cycle fluxes in general are decreased due to the presence of CAF conditioned media. And we can also look at what's happening with the utilization of glutamine. So one thing that I wanted to note here is the presence of the CAF conditioned media certainly increases the rate at which glutamine is converted into glutamate. And this mainly has to do with the fact that due to the CAF conditioned media, much more glutamine is being taken up by the cells. And we see that with the ASCT2 reaction, which is the import of glutamine from the extracellular space. Again, that's highlighted in red because that's a measured quantity that we use to constrain our model. But we see that for the dark bars, either dark blue or dark green, there's an increase in the amount of glutamine that's taken up due to the presence of the CAF conditioned media. And that certainly influences greater flux through this glutaminase reaction that converts glutamine into glutamate. Now then we see the opposite happening for cells that are remaining in CRC media. So these light bars here have a much higher rate of the glutamine synthetase reaction. So that is converting glutamate into glutamine in order to maintain the glutamine that's needed for fueling TCA cycle or for using it to produce NADPH and other metabolites that are required for cellular resources. So we can get greater insight using this constraint-based modeling approach as to how the cells are utilizing these nutrients and what are the differences that appear due to the presence of the CAF conditioned media. So to summarize, right, we have found both with looking at uptake of glucose and glutamine, but also with a detailed analysis of the flux through these central carbon metabolic pathways, that due to the presence of the CAF conditioned media, we have greater glycolysis, greater flux through the oxidative pentose phosphate pathway, greater flux through the glutaminolysis reactions, and decreased flux through TCA cycle. And these conclusions hold true for cells that have the wild type KRAS or also cells that have the KRAS mutant. So we can again get greater information about the flux through these reactions and helps us to know what are the cells relying on. Well, what carbon pathways are they relying on when they're grown in the CAF conditioned media. Then we can say, using this mathematical framework, can we predict what the effects of different gene deletions might influence? And so we can use the model to say, well, if we now set the bound for a reaction such that no flux can go through that reaction, so simulating a gene deletion, how does that influence the resulting flux distribution, but also how does that influence the growth rate? And are there particular deletions that we can predict that would have a stronger effect on cells that were switched to CAF conditioned media? We're really trying to understand how we might be able to exploit this metabolic interaction between CAFs and colorectal cancer cells, and say that if we know that now the colorectal cancer cells are more reliant on glycolysis and more reliant on the oxidative pentose phosphate pathway arm, can we exploit those vulnerabilities and see, are there certain gene deletions or enzyme knockouts that would be having a stronger effect on cells that were switched to CAF conditioned media? And the short answer is yes. So what we are able to identify are particular knockouts, enzyme knockouts or gene deletions, where the cells that were switched to CAF conditioned media are more vulnerable to those metabolic perturbations. And so anything that is right along this 45-degree angle line means that cells that were in CAF conditioned media or cells that were in remaining in colorectal cancer media, they have the same, those gene deletions have the same effect on those particular experimental conditions. But the points that are below the 45-degree angle line means that the cells that were switched to CAF conditioned media are more vulnerable to those enzyme knockouts, meaning that their growth rate is restricted more so because they've been switched to CAF conditioned media for the same enzyme knockout that we would have for cells that were remaining in CRC only media. This holds true for wild type cells. So we see a collection of reactions that if we knock out those enzymes or simulate the gene deletion, that we would have greater effect on cells that were switched to CAF conditioned media. And most of these are in glycolysis or in the pentose phosphate pathway because we know that the cells are more reliant on those reactions. So this is true for wild type cells. We have very similar results for the KRAS mutant cells, and it just helps us to see, using this mathematical framework, how we might be able to exploit those metabolic vulnerabilities. So what we're doing next is to be able to experimentally test and validate some of the model predictions. So the summary from this part of the talk is that we can look at the effect of CAF conditioned media. We see that it has a stronger impact on flux through the central carbon metabolic pathways than the KRAS mutation. We can use the model to be able to identify gene deletions that particularly affect the cells that were switched to CAF conditioned media. So taking advantage of those metabolic vulnerabilities because of the presence of CAF conditioned media. And I didn't have time to show today, but we can also look at how the CAFs promote certain metabolic alterations that also affect energy production. So we've looked at flux through the reactions, but also production of NADH, NADPH, and theoretical maximal production of ATP. And we see that CAFs also influence the production of those species. So I sort of gave a hint as to what we're doing next, still focusing on metabolic reprogramming, but we're excited to work with our collaborators in Shannon Mumenthaler's lab to validate the effects of the CAF conditioned media and those metabolic vulnerabilities. We can also look at what's happening in the reverse direction. So everything that I've talked to you about so far has been how do CAFs influence CRC cells. We can also look at how colorectal cancer cells influence CAFs. So we're looking in the reverse direction as well. And all together, we'll be able to put these constraint-based models into an agent-based model called PhysiCell. And this is with Paul Macklin's lab at Indiana University in order to not only model what's happening for a single cell population, but what's happening for a population of colorectal cancer cells that are grown together with CAFs, where the intracellular metabolic programming is really driving the cell behavior so that we can look at the effects of targeting metabolism on the population level growth. And then ultimately, we can validate our predictions using the 3D patient-derived tumor organoid model, again from Shannon's lab. So there's a really tight integration between mathematical modeling and experimental techniques that we ultimately want to use to see how can we exploit and target metabolic interactions between stromal cells, such as CAFs, and colorectal cancer cells. And this is, again, illustrating sort of that iteration and integration of experiments and mathematical modeling. So I talked a lot so far about cancer metabolism. I want to spend some time also telling you about some of the work in studying cell-cell interactions. And again, having a mathematical approach and a computational approach to this is quite useful because of the many cell types that are present in the tumor microenvironment and the different ways that they interact with one another. So here's just another way to look at it. We certainly know that there are interactions between tumor cells and immune cells. For example, T cells can become activated and promote tumor cell death. There can be interactions between different immune cell subtypes. So a naive macrophage can go towards an M2, which is a tumor promoting phenotype, or an M1, which is an anti-tumor phenotype. And really there's a there's a scale in between. It's not just these two broad classes, but also T cells can influence that differentiation. So those are immune cell interactions. And then, as I already talked about, have looked at tumor stromal interactions with the effect of CAFs, but there are other stromal cells such as endothelial cells that promote angiogenesis, and tumor cells can actually promote angiogenesis as well by secreting factors like vascular endothelial growth factor. So we've gotten really excited and interested in the interactions between cells and want to look at a modeling approach that allows us to see, well, how many cells are there? How are they interacting with one another? And how does that give rise to a spatial distribution of cells in the tumor microenvironment? So we've looked at combining the kind of mechanistic detailed model that I presented already at the metabolism level and at the cell signaling level with data-driven modeling, as well as agent-based modeling to predict tumor growth and really have a quantitative framework that allows us to explore different treatment strategies. So this is some work that I'll present now from a very talented graduate student in the lab, Colin Sess, where we've been really excited to model the tumor ecosystem. First, starting with a paper a few years ago, looking at the effect of macrophage-based therapies. And then the work that I'll talk to you about today is the evolution of tumor phenotype. So actually in this example, we're taking out the immune cells and now just looking at how tumor cells interact with one another and how their cell state evolves over time and evolves in response to the tumor microenvironment. This is, again, an agent-based model, and then we can better understand the behavior of tumor cells and then bring back in T cells and macrophages and B cells and natural killer cells. Okay, so I'll talk to you about the evolution of tumor phenotypes, particularly in response to anti-angiogenic therapy as well as chemotherapy. So we're, again, interested in modeling the tumor microenvironment, which we know influences tumor cell state. So we can think about a base tumor state evolving to different cell states based on the impact of the tumor microenvironment. So for example, the buildup of acidity or the lowering of the pH in the tumor microenvironment leads to evolution of cells tumor cells such that they gain a resistance to those low pH levels. So they have this acid resistance that makes them more robust, more migratory. We can also look at the effect of hypoxia, which is having low oxygen levels, which is also contributed to and influenced by angiogenesis. That can lead to cells having increased glycolysis, which again, has a nice handshake to the first part of the talk, but also increased migratory ability. So we can look at the effect of certain microenvironmental conditions and how that influences the tumor cell state. So I want to talk a little bit about the modeling framework that we have developed. It's agent-based model. It's different from the constraint-based metabolic model that I presented for the first part of the talk. So I think it's worth spending a little bit of time just explaining this kind of modeling approach. So we can see the evolution of a base tumor cell state into a state that has higher acid resistance, increased glycolysis, and or increased migratory ability. So these are three different characteristics of the tumor cells that we can see evolve over time and in response to the microenvironment. The cells can also proliferate. They can migrate and move around in the tumor microenvironment. They can die due to necrosis. They can also metabolize nutrients. So we've sort of boiled down almost everything that I talked about in the first part of the seminar to just look at how cells will take up oxygen, take up glucose, and secrete lactic acid in the form of hydrogen ion. And so we're really just now trying to look at what's happening at the cell level. How does it evolve to these different states and have these different features? And how does it perform these different behaviors? So talking about the modeling framework, we're using an agent-based model. It's an off-lattice center-based model, which just means that we're not forcing these cells to be in defined grid spaces. They are represented, the cells are represented with a point and a radius that determines how far, their influence, their sphere of influence is. Cells can exert forces on one another, and the surrounding tissue can exert forces on the cells as well. In addition to the cells, which are discrete agents, we also have diffusible factors that are described by partial differential equations. So we want to describe how the presence of oxygen and glucose and the hydrogen ion is varying in space. So we have partial differential equations that describe the level and the spatial distribution of these metabolic species, of angiogenic factors such as vascular endothelial growth factor or VEGF, as well as the chemotherapeutic drug that I'll introduce in a couple of slides. So we do model the tumor vasculature in this agent-based model, but it's very simple. We have blood vessels that are represented just as a point source. So we're not interested in the morphology of the blood vessels or the blood flow through the vessels. We're just interested in having them be a source of nutrients such as glucose and oxygen, and also a way to remove or take up lactate to remove the hydrogen ion from the local microenvironment. The tumor vasculature, so every point source, every vessel has a particular characteristic that we call health. And they start off as healthy, but then due to the presence of cancer cells and other microenvironmental factors, that health characteristic can decline over time. And then the vessels are recruited in response to the local VEGF concentration. So the model also accounts for host tissue, which takes up nutrients and provides this competition for the availability of the nutrients. And then just one more slide on the modeling framework. We are interested in applying this model to simulate different drug treatment strategies, namely the effect of anti-angiogenic treatment, which aims to reduce the rate at which vessels are recruited and also increase the health of the vessels, because there's been a lot of data that's shown that anti-angiogenic treatment can improve perfusion in the tumor and increase the health of the vessels themselves. And then we have chemotherapeutic agent, which induces drug damage and leads to apoptosis of the tumor cells. So we have this modeling framework. Now we can use it to answer different kinds of questions. First, what are the dynamics of tumor growth? So how many tumor cells do we have over time? And also, how do the cells evolve in response to treatment? Not only the number of cells, but what is the spatial organization and phenotype and morphology of the tumor as a whole? So I'll show you some results. We can predict the growth of tumor and also the evolution of the cell state. And so we can do this for different tumor microenvironmental conditions. So we can have a condition where there's a low recruitment of vessels, so low recruitment, and the vessels that are recruited have a low health. And we can see how the number of cells grows over time, but also how the state of the cell evolves over time. So I'm just showing you one snapshot from the end of a 200-day simulation, where we can also see how the cells go from this base phenotype, which is in gray here. Base phenotype is in gray, and evolve to either have higher glycolytic shift, to have higher acid resistance, and also to have higher migratory ability. The black points here indicate necrotic cells, so cells that are dying, and the red points are the blood vessels, those point sources. So we can try to see how does the tumor microenvironment influence the evolution of the cells and also the morphology of the tumor as a whole. So if we now go from a condition of low recruitment and low health to having low recruitment and high health, we can see a difference in the morphology overall of the tumor, namely that the tumor cells remain closer to the center. There's still this necrotic core in the center, but there's less migration away from the center of the tumor. the cells that have this higher acid resistance are now retained more so in the tumor rather than being able to move out from the center of the tumor. So just changing the health of the vessel influences the morphology of the tumor that the model predicts. We can also look at what happens when you have high recruitment rate, either for low health vessels or vessels that have high health. And this further demonstrates how the cells that are evolved to have greater glycolysis or greater acid resistance or higher migratory ability, even though they have this higher migratory ability because of the high recruitment of the vessels, that really constrains the cells and retains them in this circular morphology. We do see some areas of necrosis, but again, the cells are retained inside of the tumor rather than being able to migrate out. So just understanding the evolution of the cells and how the tumor grows when we have different tumor microenvironmental conditions. Now we can take a step forward and say, well, what happens in response to anti-angiogenic treatment? And we can see now if we just stick with one environmental condition, which is having high vessel recruitment and low vessel health, we can see again what happens without any treatment at all. The cells are quite contained in this circular morphology. But when we add anti-angiogenic treatment, it certainly has the effect of reducing formation of blood vessels. So fewer blood vessels are being present here. Certainly in the center of the tumor, we don't have many of the red dots there. But it has this side effect of making the cells evolve into having greater reliance on glycolysis and greater acid resistance and a higher migratory ability. And this has also, been seen in preclinical models where the effect of the anti-angiogenic treatment can have this counterintuitive impact to actually have more aggressive and larger tumors, and then cells that have greater migratory ability as well. So it certainly influences the recruitment of blood vessels, but it has this effect of allowing the cells to evolve and move out of the center of the tumor into the surrounding local microenvironment. And we can also quantify this movement out of the center of the tumor, where if we look at the frequency of cells, so how many cells are present in a certain distance from the tumor center, we can see what happens when we have control, so no treatment, and how that distribution is really shifted towards the right, meaning that the cells are moving away from the center of the center of the tumor when we have anti-angiogenic treatment. So a quantitative way to see this qualitative result of greater dispersion of the tumor cells. And then we can also look at what happens when we add chemotherapy. So combining anti-angiogenic therapy with chemotherapy. Chemotherapy alone, we see what happens, and that's shown in blue for different levels of resistance to chemo. So we can allow the cells to have some tolerance to chemotherapy. And as we increase that tolerance, we certainly see that the tumor growth takes off. And then we can see that adding anti-angiogenic therapy in combination with chemotherapy does limit the growth of the tumors. But it also leads to having higher acid resistance, less glycolysis, less migratory effect when we add anti-angiogenic treatment on top of chemotherapy, but the cells are more resistant to acid, so they're more robust to the low pH levels. And then one last set of results here to show not just the growth curves, but also the morphology and the spatial organization of the tumor when we have chemotherapeutic agent provided alone or chemotherapeutic agent administered alongside of anti-angiogenic agent. And so we can again look at what happens when we increase the resistance to chemotherapy and see how that changes the morphology. Again, I'm now just showing the state of the cells in terms of their migratory ability, but we can also look at the resistance to acid, low pH levels, and also the glycolytic shift as well. But again, we see that even adding chemotherapy together with anti-angiogenic therapy, we still see this increase in the migratory ability of the cells compared to chemotherapy alone, where we have that evolution to a higher migratory state, but they're retained in the center of the tumor. And again, we can look at what happens more quantitatively to see the distribution of the cells and how far they are from the tumor center with anti-angiogenic therapy alone, with chemotherapy alone in blue, and then with the combination. So just to summarize the second part of the talk, I presented now an agent-based model that helps us to predict how cancer cell state evolves with time in a heterogeneous tumor microenvironment. We can look at the effect of anti-angiogenic treatment and how that allows the cell to evolve towards more of a glycolytic phenotype, more invasive, and with higher acid resistance. And then combining anti-angiogenic treatment with chemotherapy does alleviate this harsh microenvironment, meaning the microenvironment that would cause the cells to evolve. They have less pressure to adapt, so it's helpful to chemotherapy, but we still see this invasive phenotype in terms of cells moving away from the center of the tumor, certainly compared to no treatment at all, and also in comparison to chemotherapy alone. So what's next? We're interested in validating some of the model predictions, again, with 3D patient-derived tumor organoids. We're interested also in calibrating the agent-based model to data from tumor images. So you might have been mindful or seen that I haven't said that this agent-based model is not specifically applied to a particular cancer type. And this is because we haven't really calibrated it to be cancer type specific. And now we're working with Dr. Ivanthia Roussos-Torres at the Keck School of Medicine here at USC to be able to calibrate the agent-based model to imaging data that she is collecting for breast cancer patients. And we can also look further inside of the cell and use, the wealth of data in terms of single cell transcriptomics information that we can use to infer the gene regulatory network, for example, and have that be the intracellular program that's influencing the cell level decision. So really building up this multi-scale approach to studying and modeling the tumor ecosystem. So with that, I will thank and acknowledge the students in my lab, particularly a former postdoc, Jumin Wang, who was very much pushing forward the metabolism story that I told in the beginning, and also Colin Sess, who's been moving forward with the agent-based modeling. Definitely want to acknowledge our collaborators on the experimental side and also on the modeling side with the Physicell that we're moving into with the metabolism work, and acknowledge our funding sources. And thank all of you for your attention, and I'm happy to answer questions.
All right. Thank you very much, Dr. Finley. I just want to make sure we all got back on here. Wonderful talk, really enjoyed it. There's nothing in the chat, but I'm sure there's a lot of questions out there, so I'm gonna have people potentially raise their hand so I can help moderate this. So if you have a question, put your hand up. But I want to start with one myself, and I'm really curious at this last point. You're talking about ways to develop calibration tools, and I think of sort of two opportunities. One is sort of opportunities to be using higher level imaging tools, sort of digital histology methods to gain insight that could perhaps beyond just cell distribution, but something about cell state or other features within it. Whether or not that is possible to be integrated into some of these agent-based models at the end. But the question I'm mostly interested in is ways in which, like, the engineered models that we work on could be really driving some of these responses. We think about how do we put together multiple cell types and spatial organization. The thing that's always I'm curious about is, we can make models that have defined ratios and the like, but how many of those are useful to make an actionable model? When you start thinking about, you could come up with a million variations, I'm sure, but what do you need to start building actionable models of the tumor response in the end?
Yeah, so it's a great question, and something that we have been thinking about a lot, because I think that we have, a very nice framework for making some predictions. We want to make sure that the predictions are realistic and make sense, and so we have to rely on experimental data. I think that in this example that I showed, right, it's just cancer cells. And so we want to vary the tumor microenvironment. I think the kind of platforms that you have are very much minimal to that, right? You can more finely tune the tumor microenvironment and different properties in order to measure numbers of cells and even the distribution and spatial organization of cells. But also, right, like you're saying, take a snapshot and see what's the state of the cell and can we map that back to what the model is predicting. So I think we need certainly, a suite of experiments that have co-culture, different tumor microenvironmental conditions, but we also need to say, well, are we including in the model the right cell types and the right kinds of interactions? One other thing that I'm excited about the kinds of platforms that you have is that you can turn, I think, right, you can turn on and off interactions, maybe simulating or stimulating the cells with different cytokines or different growth factors to see how that influences their interactions. And that's the kind of mechanistic detail that we need to incorporate based on data, incorporate into the model.
Thank you, Stacey. I see one thing in the chat, but Eric got his hand up first, so I'll pass it to Eric.
Okay.
Yeah, thanks. Lovely talk. Great work. My question is for the first part, when you're looking at the CAFs and finding that basically they're increasing the metabolism of the cancer cells, especially like glycolysis and that sort of things. And so like cancer cells, they're understood to already have a really high glycolysis or glycolytic index. So I'm just spitballing here, but based on some of your modeling, could you, instead of, and I know you showed some work where you like knocked out some of the important enzymes and showed that would disrupt things, but could you consider doing the opposite and approach it with like a synthetic lethality approach, sort of like, all right, they're already ramped up, can we push them over the top? Similar to PARP inhibition is a synthetic lethal approach?
Yeah, I'm wondering though, so I think that would be very relevant. I'm wondering how the constraint-based model would account for such behavior, right? So constraint-based modeling, we're trying to maximize with this knockout even that I show here, right? We're trying to maximize biomass production, which is a proxy for growth.
Right.
So in this case, we want to say, well, how can we further alter the metabolic networks to promote lethality, right? So I'm trying to think about how, like, what would the objective in that case be, right? We have to define this objective function for the constraint-based modeling. So thinking about how that could be informed by data, so that we're trying to force the cell to do an appropriate behavior.
Yeah, I guess I imagine we would have to figure out at what point are they dividing so fast that relying on glycolysis is not meeting their energy needs or something like that. But
yeah. I mean,
you would identify targets though, right? In that way, you could still think about ones you could approach from that perspective.
Mm-hmm.
Maybe. Yeah, I think so. I should say, and just a reminder, that this is for just central carbon metabolism. So we're actually working right now to have a genome scale model of metabolism for colorectal cancer cells and macrophages and CAFs. And then we can further look at the interactions between these in silico cells. But because I think, right, it's going to go beyond actually glycolysis and PPP and TCA cycle.
Right. No doubt it'll touch cholesterol biology.
Of course. But it's a great question. And yeah, I think I have to just think more about what would the objective or how to formulate the problem.
I'll follow on with a quick second question if I'm allowed, Brendan.
Go for it.
In that same model system, so the CAFs that somehow are ramping up this metabolism like glycolysis. Do you have any sense from your modeling or from the literature, like how are they doing that? How are calves boosting glycolysis?
Exactly. The short answer is no, we haven't. So when we did the measurements of extracellular metabolites, it was very targeted. So we just measured glucose and glutamine and lactate. But really what we need to do is do a profile of the calf conditioned media and start to see what could the candidates be that are mediating this interaction. And right, we could do it on the metabolism level, and that's what we're, sticking our, putting our flag in. But the truth is, it could also be signaling factors and other biochemical factors that we would not account for with the kind of model that we have. But at least on the metabolism side, we're working on profiling the calf conditioned media and also looking at a newer approach that looks at predicting what are the metabolites that could be secreted by one cell type with no cost to the growth of the cell and taken up by another cell type. So somehow it's sort of simulating like consortia of cells that are growing together. It's been applied more so in like microbial consortia, and now we're applying it in cancer. It's something that I'm working on writing literally right now, so I'm super excited about it. But it tries to answer the exact question that you asked, which is, what are the factors that are mediating these interactions? Once we know those, we can have this, prioritized list and then use those for exploiting the interactions further.
Yeah, that's awesome. I yield the floor.
All right. Thank you, Eric. We have one question here in the chat. Victoria, do you want to unmute and ask it? Maybe, maybe not. I may ask it for her. So in your agent-based model, how many phenotypic states are you limited to? Can this model be extended to address specific molecular changes rather than phenotypic shifts in cells?
Right. So I didn't, I took out the slide where we have basically like the distribution of cells and the extent of, for example, the glycolytic shift or the extent of the acid resistance. But really, it's not just like on or off, right? It's a continuum of states from this base phenotype to a very high level of acid resistance. And so it's this continuum of states. We have just focused on these three because it's sort of motivated by our interest in angiogenesis and VEGF, and also our interest in the effects of metabolism, again, just boiled down to glucose uptake and secretion of lactate. But if we had more biological knowledge and data-driven insights about what are the states that we should be keeping track of, we can incorporate that into the model. And I think, right, that also allows us to take advantage of omics level profiling of the cells to define what the states are. And so that's another area of research that we're moving into. One of my postdoc now in the lab is looking at the evolution of T cell states, because we have a collaborator that's been able to profile the states of T cells, and we can use that. Really, they're looking at single cell transcriptomics as a way to characterize the state. And so it's really just dependent on what kinds of questions we're interested in answering, and also the kind of data that we have to inform what the evolution would look like.
That's good. Yeah, because that leads to the last question I had, that we have some of work in our group that we've been looking at creating cell spheroids of patient-derived specimens and looking at spatial distribution of cell subtypes within these patient specimens, and as they start to invade or respond in a tumor environment, it layers on top of it itself in terms of identifying multiple sort of phenotypic subsets we're interested in, either from a functional response or a drug targeting sense. So that, I think that's, I'm pretty sure that's where Victoria is coming from that. So that's really, really exciting. So well, we've reached the top of the hour, and I want to make sure we respect everyone's time. Stacey, it was wonderful to have you join us. Thank you for spending the morning with us, especially the early morning, seeing that you're on the West Coast, to run this through. And if folks have other questions, you feel free to follow up with Dr. Finley directly. And other than that, I want to thank you again for spending the time with us. So